Okay, so hi everyone. I hope you all saw my announcement and you all know that this lecture that I will give today will be pre-recorded. The reason why I decided to pre-record this lecture is because on Tuesday I had issues with internet connections during class after you. So in order to avoid that, like many people couldn't hear it very well, so I didn't want to torture students, and I decided to pre-record myself so I don't have those issues with internet anymore. So when I come back next week, we will continue being on in person. Everything will be as it should be. And for now, this lecture will be pre-recorded. And if something is unclear from this, you can, of course, as usual, always contact me or Brittany. Again, Brittany will be there on Thursday, so you can. She will give you an online lecture about the assignments. Unless she decides differently, I think she will be there, but I am not going to risk to get myself interrupted, so I will just pre-record my lecture. Anyway, moving on with microbial cell structure. So last class we covered we basically had a brief recap of microbial cell features. And we covered cell wall in gram-positive, negative, and archaea. And we talked about three basic motility structures that you need to know. And today we will stay with cell wall glycans and cell-shaped proteins. So in terms of cell wall glycan, the first glycan that we have to cover is, of course, peptidoglycan. It's a big component of cell wall present in both gram-negative and gram-positive. As we know, in gram-positive, it's a very thick layer. In a negative, it's very thin in between two membranes. And then we will also talk about techoic acids. As we know, there are lipotechoic and techoic acid, depending on which part of bacterial cell they bind. If they bind to the cell membrane, then they're lipotechoic acid. If they bind directly to peptidoglycan, they are just techoic acids, and techoic acids are a landmark of gram-positive bacteria. Peptidoglycan exists in both. And there is lipopolysaccharide that we will talk about, and LPS or LPS present in gram-negative bacteria, landmark of gram-negative bacteria. Now, in gram-negative bacteria, we will start with them. There is that very thin peptidoglycan layer that is in between two membranes, as shown here on picture. So it's pretty much in between outer and inner membrane. Then you will have this outer membrane structure that is characteristic of gram-negative bacteria, and you will have this lipopolysaccharide, which is labeled here. So this structure here is called LPS. Molecules in gram-positive bacteria, you will have voltechoic acids and techoic acids. So you will see lipotechoic acids here. So this top picture. So this is where this lipotechoic acid is. So again, if it's bound directed to peptidoglycan, that's called techoic acid. If it's bound to the cell membrane, it's called lipotechoic acid. So this one is called lipotechoic acid, and there is techoic acid here, which you can see bound directly to peptidoglycan. Now, all these molecules are synthesized in cytoplasm, and then they have to be mobilized into the periplasmic space in order to build all those glycan structures. The main molecule in bacterial cell that is involved in transporting all these molecules from cytoplasm across the membrane and into the periplasm is called flippase. And this is an important molecule that we will discuss in details. So the flippase is an enzyme, the name tells you, that attaches, actually, technically, it is not an enzyme, so let's stay on that. I always get distracted with the name. So it does have a function of enzyme, but it's actually not an enzyme. So I have to be careful what I'm telling you in terms of those things. So this flippase attaches the monomers in the cytoplasmic space over the membrane. So basically, flippase is attaching monomers in cytosol, basically on cytoplasmic side of the membrane, and then literally flips it over to periplasmic side. So, and this molecule is super important in building bacterial cell envelope. The molecule is called undecaprenyl phosphate or bactoprenyl phosphate shortly. it's enough to say bactoprenyl phosphate. This is an essential molecule, and it's involved not only in synthesis of peptidoglycan, but also in synthesis of lipopolysaccharides, exopolysaccharides, tehoic acid, glycosylation in some bacteria. So basically, this would be analog to dolichyl phosphate in eukaryotes, and it's used to translocate sugars across inner bacterial membrane. So bactoprenyl phosphate is a molecule that you need to memorize. And now you will see why bactoprenyl phosphate matters. So in terms of peptidoglycan biosynthesis, this is the process, the first important process that I will ask you to understand and to know, among many others, especially in second part of the class, but this one will be the first. So peptidoglycan biosynthesis is broadly divided in two events that are happening in cytoplasm and in periplasm. And here order actually matters. Now, in what's happening in cytoplasm? So in cytoplasm, first you will see the synthesis of monomers that are required to build peptidoglycan. So those monomers are NAG and NAM, so these are short names for N-acetylglucosamine. So that one is dominant monomer, and N-acetylmuramic acid is for NAM. Now, so NAG is a dominant molecule inside cytoplasm. Then NAG is linked to the UDP, uracil nucleotide diphosphate, in cytosol. So build NAG, bind to UDP. Then you have this complex NAG-UDP, and then you have phosphoenol pyruvate molecule, which is this PEP. And when phosphoenol pyruvate molecule is hydrolyzed, energy is released. That phosphoenol pyruvate has high energy bonds. Now, the energy from that PEP hydrolysis is used to attach 3 carbon atoms to NAG. Remember, NAG is now bound to UDP. That reduces double bond, and that's how you get NAM. So basically, the point is that before you start this synthesis of peptidoglycan, you have to build monomers required for it. Monomers are NAG and NAM. NAG is dominant monomer, and NAG is used to make NAM. How you make NAM? NAG, UDP, PEP hydrolysis, NAM. So again, I know it's an intensive process, but we can practice. Then in step 2, you will have NAM peptidation, which is happening in cytoplasm. So basically, you have 3 peptides that will be added to the built NAM. Again, you build NAM from NAG. Then you will have 2 peptides addition, and those 2 peptides are known as diala conserved motif, and those 2 are very important for antibiotic recognition. We will cover that in the next few slides. So diala motif, 2 alanines in this structure. So again, step 1, synthesis of monomers for biosynthesis. So you have NAG, you have NAM. Step 2, add 3 peptides that are species specific, so they are not conserved, not like diala. So peptidation of NAM, so NAM plus 3, then you have addition of 2 diala, conserved motif, and then all that NAM pentapeptide, so NAM plus 3 plus 5 is transferred to this bactoprenyl phosphate. Then, so when you have this event completed, you get this NAG being attached to NAM pentapeptide bactoprenyl. So even though you first had to have NAG built, so now you have basically all that structure, NAM pentapeptide bactoprenyl NAG, and now autolysins are enzymes that are basically mediating cell wall opening. So they're opening, those are molecules that are opening cell wall, and they create space for insertion of new monomers. So those autolysins are now mediating the cell wall opening, and so autolysins are molecules that are essentially enzymes, so amidases, glucosidases, or peptidases. So they're breaking chemical bonds in cell wall to make a space so new monomers can be inserted. And autolysins have to be tightly regulated, because if they are not, they can cause literally suicidal activity of the cell. So again, now when autolysin is made a space, bactoprenol is the one that mediates flipping of NAG-NAM pentapeptide over cell membrane. Bactoprenol stays inside. Now you have periplasmic events where you have all those components required to build peptidoglycan of cell. So in the next step, that's happening in periplasm, you have formation of glycosylate peptide bonds in periplasm. So there are two enzymes involved in that. Transglycosylase forms enzymes between NAG and NAM, so basically two sugars, transglycosylation, glycosyl, sugar, transglycosylation. Another one, transpeptidase, forms peptide bond between two alanines, so it makes that diala-diala conserved motif. And there is a very cool video of peptidoglycan biosynthesis that you can check if you want. Now, why this transpeptidation in periplasm is important? Well, because it's antibiotic target. So maybe some of you heard about this. So penicillin is very famous antibiotic. I'm sure you all know about penicillin. So the mechanism of penicillin action is actually to mimic this conserved diala-diala motif. So they look quite similar. Penicillin, diala-diala. They're quite similar, which means they can trick the cell. So when bacteria are actively growing and they're building that cell wall, this enzyme transpeptidase will create the bond between diala-diala, so make this conserved motif, linking them together. So there is a peptide bond. So the way how penicillin will damage bacteria is it actually tricks cells, so it mimics diala-diala motif. So transpeptidase thinks that this is a diala-diala motif, but it's actually penicillin, so it gets damaged and then cannot complete cross-linkage of cell wall monomers, so bacteria cannot grow. So penicillin belongs to beta-lactam antibiotics. So why it's called beta-lactam, and you can see picture here because it contains this beta-lactam ring. So since beta-lactams are analogs of diala-diala, transpeptidase binds them. So that's basically penicillin tricks transpeptidase. So when transpeptidase cleaves that beta-lactam ring, like thinking that, oh God, this is my diala-diala, so it cleaves it, all enzyme remains covalently attached to the ring itself. So then basically that beta-lactam ring blocks transpeptidase. So transpeptidase is trapped and cell wall synthesis cannot be performed. So the way how cell get resistance to penicillin, so basically the way how it happens is when bacteria adopts enzyme beta-lactamase, so it can degrade beta-lactam ring in penicillin. Now, in terms of vancomycin, make sure to understand vancomycin and penicillin are not same type of antibiotics. So they do have same target in bacterial cell. They all act on transpeptidase, but they have different mechanisms. So vancomycin literally covers diala-diala motif. And when it's covered, transpeptidase, so masking it, so transpeptidase cannot recognize it. So basically, it's a same principle, so it affects diala-diala, but it's not the same mechanism of action. And beta-lactam's penicillin is beta-lactam antibiotic, while vancomycin is not. Now, bacteria can develop vancomycin resistance. So how? So if bacteria has a normal growth, it has normal diala motif, vancomycin will bind to these precursors and block incorporation. So it's sensitive. But if some mutation happens, so instead of diala, you have diala-dialac, like here, that then vancomycin won't be able to cover it. So this is all about peptidoglycan. In terms of voltaic acids in gram-positive bacteria, there are a few interesting information that I wanted to share with you. So first of all, they are gram-positive specific landmark, specifically in gram-positive bacteria. You do need to understand that those are polymers made of glycerol and ribitol. those polymers are joined by a phosphate group, but it's important to understand that you have ribitol and glycerol moiety and various sugars attached to those moieties. So basically sugars or amino acids can be attached to those glycerols or ribitols, and then all that structure is covalently attached either to plasma membrane or to peptidoglycan. If it's directly attached to peptidoglycan, it is voltejoic acid, WTA, but if it's attached to the plasma membrane, it's lipotejoic acid. There are various proposed roles of these tejoic acids in a gram-positive cell, and there are a few controversies about that. So for now, I will leave you with these few functions. So they participate in cell division, ion homeostasis, control of autolysins, pathogen bindings, and biofilm formation. I did not find any information about tejoic acid being immunogenic. Again, maybe it's possible that I just didn't find it. So if somebody has information, feel free to share. Next one is LPS or lipopolysaccharide in gram-negative bacteria. So those are very important cell-specific gram-negative structure. So the structure of all LPS is something that I will insist on. So it is very important to know, and we mentioned previous class, if you remember, lipid A, core polysaccharide, O antigen. So basically, the structure that is exposed on the top of outer membrane is called LPS or lipopolysaccharide. So it is unique and conservative structure in gram-negative bacteria. And many bacterial strains and diversity among bacterial strains is formed because of variations of specific glycans present in lipopolysaccharide. So what we need to know about LPS, and again, this is quite important, and I did mention also previous time. So three main structures in LPS, and they are on the top of the cell membrane. So what you have is lipid A that comes first, if you look from cytosol toward the outside. So lipid A is structure that contains fatty acids. So you see it here, chains of fatty acid, and those fatty acids get stuck into the cell membrane. So this is basically on the cell membrane. Okay, hydrophobic, of course. Then you have this core polysaccharide. So inner and outer, it's enough to understand it's a polysaccharide core. So this structure is joined to the lipid A. And there are sugars over there. And third is O antigen. This is strictly polysaccharide chain that is extended from the core, and it has various roles. So O antigen is what actually distinguishes serotype of different types of pathogens. So that O antigen is quite important. So what are roles of antigen? So it participates in charges of the cell. It stabilizes membrane structure, attachment of the biofilm formation. It decides what will come in and out, so permeability barrier, protects from host defense. So what O antigen does, it elicits immune response, and very often O antigen is changing so it can trick host defense. So basically, this O antigen is highly important for immune system. Another specific structure is LOS, lipo-oligosaccharide. So it has exactly the same structure as LPS, just without O antigens, but it also is present in pathogenic strains, Bordetella, Neisseria, Campylobacter. Probably the most famous is Campylobacter. So both LPS and LOS are synthesized on cytoplasmic side of the membrane, and the principle of synthesis is not too different. I mean, it is, but you can find the similarities between the synthesis of this glycan and peptidoglycan. So again, there is the role of bactoprenol. So via bactoprenol phosphate, all this structure will be transferred in periplasmic space, so very similar like peptidoglycan. But then in periplasmic space, molecules are moving using ATP-derived energy because they have to reach the top of the membrane. So again, this mechanism is not always bactoprenol mediated. So this is the difference between peptidoglycan and this type of glycan. And we will learn about few pathways that are important for LPS synthesis. So there is this LPT pathway for LPT transfer, and let's go through this briefly. So there is this MSBA cassette. So this is a protein, a receptor that can bind ATP. So this MSBA, this is transporter. So it actually does flipping of LPS from cytoplasmic membrane from cytoplasmic to periplasmic space. So, and it's shown here in the picture. So you see how it binds ATP, and it has ATP hydrolytic activity. So basically LPS would remain then in inner membrane. And then this LPTB2FG, of course, I won't ask you to memorize this. So this transporter completes ATP hydrolysis, and that energy is used to extract LPS from lipid membrane. So what do you have basically is that LPS is first made in cytoplasm, okay? And then, thanks to this MSBA, LPS is bound. So basically, LPS is binding to this MSBA. So MSBA, so this is how it goes. So LPS stays in inner membrane. Then it binds to MSBA. Then there is hydrolytic activity of another enzyme, so ATP is degraded, so energy is released, and that energy is used to actually extract LPS from the membrane. But this MSBA is very important for the docking. Then you have another molecule called LPTC that interacts with LPTB, and that interaction, so when they interact, they literally take over this LPS that was previously made. So basically, you can see that coordinating function between MSBA, LPT, B, and C is taking over LPS and driving into the inner membrane all the way down to the periplasmic space. So in periplasmic space, LPS moves further. There are other proteins that participate in that. So there is protein A that is in peptidoglycan, protein D. So all these LPT proteins participate in a binding and transport of LPS from cytoplasmic through periplasmic to periplasmic space. But the most important molecule here is MSBA. So MSBA mediates the transfer of LPS from cytoplasmic to periplasmic space, but this is not bactoprenol phosphate. Second one, probably more famous, is KDO pathway for LPS transfer. And there are a few steps that are very important, and I will emphasize them. So as you probably remember, because we just talked about that, there is that NAG molecule in cytoplasm, the part of peptidoglycan, and that NAG has other roles as well, not only in peptidoglycan synthesis. So NAG is simultaneously acetylated and deacetylated, and basically you have that constant deacetylation, acetylation, and in the end, from NAG, you get lipid A tetraacyl. So basically addition, removal of acyl groups, and at the end, you have 4 of acyl groups stuck to the molecule called lipid A. Now, there is KDO transporter that is transferred to the lipid A moiety, and there is an enzyme that does that. It's called transferase. And now the new round of acetylation is done. So you have lipid A tetraacyl, then addition of KDO transporter. Now you have KDO lipid A tetraacyl, and then you have another set of acetylations, and then you have glycosylation, and all happens on this KDO lipid A moiety. The next step is the attachment of this KDO lipid A moiety to the MSBA transporter. Again, you have this MSBA, so quite important mediator into the LPS synthesis. And again, MSBA mediates this transport, which we saw in the similar manner like here, through inner membrane, periplasmic space, and outer membrane. Now, simultaneously, all antigen units are synthesized on cytoplasmic side of the membrane. So I forgot to mention, so this with KDO transporter, you will get assembled lipidol and the core polysaccharide, but all antigen units are assembled separately. So basically at the same time, all antigen units, the one that goes on the far top, are synthesized on cytoplasmic side of the membrane. They are attached to bactoprenol and flipped over the membrane. So in this case, you will have and bactoprenol and MSBA mediating the transfer of two parts of the same unit, LPS. At the end, ligase called Vol catalyzed ligation of O antigen to and oligosaccharide core, but that happens outside, like on periplasmic side of the membrane. And there are some interesting LPS features that I just wanted to share with you. There are many papers published on these topics. So LPS is highly immunogenic. So unlike for tejoic acids, for this one, we know that it's very immunogenic. And it's the reason why it's so immunogenic is because of the lipid A, which is known as endotoxin. So when endotoxin is present in mammalian cells, LPS attaches to the macrophages and initiates immune response. So very often mammals can get septic shock as the organism is trying to clear the infection. So it's really highly immunogenic. And E. coli gram-negative bacteria, often used as microbial factory to produce something that will be used in humans or animals. So companies are putting a lot of effort to remove this endotoxin because it's so immunogenic that if they don't remove it and it accidentally shows up in production strain, company is done. And in terms of other cellular glycans, I will just briefly mention exopolysaccharides. So they build capsules or mucous layers that protects bacteria against dehydration. And biosynthesis of exopolysaccharides follows the similar principles as LPS and peptidoglycan units. So units are assembled on cytoplasmic sides, flip over with a flipase, and build complex structure out of the cell. And the other glycan that I wanted to mention is archaeal glycoprotein S layer that we talked about last class. And the last topic for microbial cell features are cell-shaped proteins. So there are three of them that we will cover, and they are involved in formation of different morphologies of bacterial cell. Again, there are only three of them. So those proteins are analog to the eukaryotic cytoskeletal proteins, actin, tubulin, and intermediate filaments. And you do need to know what is analog to what. So MREB is homolog of actin, FTSZ is homolog of tubulin, and the last but not least, crescentin is homolog to intermediate filaments. And the other cell-shaped proteins that are very important are penicillin binding proteins, and these PBPs are causing a lot of issues with students. So many times students don't understand. So if you are not sure, please ask me or Brittany. Now, what is FTSZ? So as we said, this is homolog of tubulin. If we talk about the eukaryotic cytoskeleton. So this protein participates in cellular division and formation of septum. This protein will be covered in details in Unit 2 when we talk about physiology of bacterial growth. Another thing that another protein that you need to know is MREB, which is structural protein and anchor. So PBP is actin homolog, so we talked about that here. So MREB is actin, FTSZ is tubulin homolog, and it is MREB is interacting with penicillin binding proteins. So it's helping in localization and control of cell shape. So you can see here how cells look when they have normal MREB and when they don't have MREB. So basically MREB is important for cell shape, so for determination of cell shape. Crescentin is homolog to intermediate filaments. So it's basically using MICRB to attach, and it is very well studied in this Caulobacter bacteria, which is bacteria that has stalk, and there are many papers published, and we will talk about some of them later in the course. So this crescentin, as you can see, goes all through the cell. So basically, if you have same like intermediate filaments, so these intermediate filaments are connecting those actins and tubulins. So the same is here. Basically, FTSG, MICRB are analog to cytoskeletal proteins, and crescentin is connecting them. It has high role in mechanical stress. So basically, crescentin filament interacts with MICRB protein to locally inhibit cell wall synthesis and induce the curved cell shape. So this is what gives cell shape to Caulobacter. And penicillin binding proteins, or PBPs, are important class of shape determining proteins that have a function to cross-link FTSG, MICRB, and crescentin to peptidoglycan. And they also cross-link peptidoglycan in the cell wall. So if you look at the list of these penicillin binding protein, you will see transglycosylase and transpeptidase. Remember, those are enzymes that are operating in periplasm during peptidoglycan biosynthesis. So again, penicillin binding proteins are participating in cell shape. So don't be confused with that. There are many confusions. Again, I cannot tell you what, because there will be questions about that, but think about that also transpeptidase and transglycosylase are considered penicillin binding proteins. And this was short lecture, so this would be all from me for now. I will stop recording. I will upload this on eClass, and we will see each other on Tuesday at 11 when I come back, if I come safely. See you soon. I'm looking forward to our in-person class finally.